to our seminar on open science organized by the Italian Reproducibility Network. This is the 7th iteration of this initiative, and I'm going to introduce you the speaker in a few seconds. But first, a little bit of housekeeping. I want to thank the AIP, Italian Association of Psychology, that is supporting these seminars by hosting them on their Zoom account. Second, most of you are already members of ITRN, but for those of you who are not, please join the network, subscribing to the link indicated below. It's itrn.org/community. It's free, you don't have to pay. It allows you to join our interesting and lively community, of course, and stay up to date with our activities. And it allows the network to grow stronger and stronger, and to enable other initiatives like this in the next months and years. Also, I would remind you to subscribe to our YouTube channel, where we have posted the recordings of these seminars. It would come online in a few days, but in the meantime, you can browse around and see the previous initiatives in these cities. Regarding our next activities, the meeting in Florence is almost here. It will be held next week, Friday, 13th of May. There are still places for in-person attending, but you can also follow us online. We're also preparing our first hands-on seminar on pre-registration for next month. We will disseminate information about this hands-on tutorial in the next few weeks. So keep an eye on either the mailing list or the website. Finally, you probably already know this, but reading doesn't work. We will have a question session at the end of the seminar. As usual, we will give priority on questions by doctoral students, and Dr. Federica Conte will moderate the questions and the answer session. We'll find in the chat some info about how it's organized. So I think this would end the housekeeping session, and I'm going to introduce the speaker. Today we're glad to introduce Marco Perugini, Professor of Psychometrics at University of Milano Bicocca. Marco's research is on a wide range of topics spanning different areas, for example, attitudes, personality, social cognition, and so on and so forth. One can safely say that between Marco and open science practices, it was love at first sight. We remember that it was about year 2010 when he was already speaking about the importance of pre-registering study, even before pre-registration was a thing. He's been involved in a number of recent contributions related to reproducibility of scientific findings, and he also wrote many interesting and useful papers on robust design and power analysis. The signal and the noise, sound research design, and power analysis. Professor Perugini, the floor is yours.
Much for the kind introduction. Thank you for having me here. I shall share my slides. I think you can see them, right? Everything okay? Great. Okay, we start. And I try to focus basically. I mean, the main point of today is going to. Sorry, the second part. But I'm going to say a few things also about the first part for replicability. I mean, it's something like almost trivial in this context, because I'm sure that you already know even more than I do about all this stuff. But just to refresh a few things why we are here to start with. I mean, what is the basic issue? Why there is a problem in replicability? Once again, I'll be very quick. But just to remind you that 2011 has been a year which has somehow made a difference in terms of what happened before, what happened after that. And there have been a few events that have really signed the field. BEM is the non-extrasensorial perception study, which basically the bottom line is, to make it short, is basically it published in one of the top journals in the field, Journal of Applied Social Psychology, a series of studies showing that things happen before they do, in the sense of causality as a reverse engineering, you first are assigned, for instance, one of the examples of the condition, you first are recalling words, and then after you assign to the experimental condition to rehearse words, and you perform better. Even if you are being assigned to the training condition after the dependent variable. Wow, that's really something like, you are inverting the law of physics, basically. And that was published in the top journal. So that created a kind of series of reactions, different types. One of the reactions, some people initially, they tried to replicate the study, they couldn't manage to, and their papers was rejected by the journal, by the editor of the journal, saying, we are not interested in replication. That was pretty bad stuff at the days, I mean, pretty bad reaction those days. And eventually, there was a publication of failed replication there, but this really opened up the issue, okay, what's wrong with the way we analyze the first, why replication, there is no space for replication stuff. First. Second, what's wrong with how we analyze data? How is it possible that some kind of data like this, they show evidence in favor of something that we know is unlikely to be correct, and replication studies have shown that it's not correct. How comes? And this is kind of replication study showing that basically there is no effect in there. Then there was a case of resignation because of invented data, basically, and basically studies published, papers published based on fake data. Again, big outcry, big reactions, but the key point was how comes no one spotted this before? It took so many years to understand that there was a problem there, even though on hindsight it was clear that there was something wrong with the data that he published. But no one, I mean, it was not catched. It was not catched. So the point is outcomes, again, strong reaction. And then on the other side, showing that basically with some kind of degrees of flexibility, you can provide evidence for anything to become seen. I'm going to the details, only to say that this is at a very huge scientific impact. This paper is one of the most studied papers since its publication today. Just to say that the field was kind of ready for this kind of emphasis on what's wrong and why things should be changed. So that's the so-called replicability. So, all this stuff, and that there's been an increasing appreciation of the problems, and especially the most important thing in my view is that there have been many, many positive improvements, many positive steps in the field. There have been changes in the standard for research for publishing. The main concept that before was swept almost under the carpet, one main concept that came out was the concept of replicability. That there being, this is a key concept in science, and this is one of the distinctions between scientific knowledge and ideology or religion or other types of approach to knowledge, which is show me the data. Basically, if I do something under certain condition, scientifically speaking, this result should be replicated given the condition. If it's not replicated, there is a problem. That's the kind of very simple basic idea coming out of centuries of time. And it has been somehow lost in the last decades, and now it's back in the forefront. And of course, replication doesn't mean validity. You can have something replicated, but not necessarily valid. But again, it's the precondition. If you can't even replicate the effect, how can it be valid? That's the kind of logic. Yeah. Now, what is replicability? And I mean, ironically, now it's clearer than it was before in terms, I mean, conceptually okay, but in terms of technology, because basically now there is a distinction, a clear-cut distinction between reproducibility and replicability. And this recent book is, I think, is a nice reference in this sense. It's also interesting. It's published by the National Academy in USA of Science, Engineering, and Medicine. It's not psychology. See what I mean? It's beyond psychology. It's a big impact of psychology in science. And the point is, now there is a clear distinction between reproducibility and replicability. The irony is that one of the most important contribution to replicability has been called the reproducibility project. So it's been called with a wrong name. In 2015, there was not clear that the name could have been distinguished. that way as it is today. Reproducible means that you can do the same results. For instance, you can have reproducible code, reproducible results. It's an internal thing. And even then, it's not obvious. There are many things that are difficult to reproduce. Replicate means consistent results with different styles. So that's the difference between reproducibility and replicability. And first, to be replicable, something needs to be described in a way such that can be replicated. Now, right or not right. I mean, any one of you who has tried to replicate a study will know by first experience that you can't do that unless you contact the main. There are so many details, most of the time. I mean, now things are a bit changed, but there are so many details that are not part of the publication. The description of the method is very short. It doesn't cover all the possible details that are part of the study. Some of them actually are quite important, and they're not necessarily part of the things that are shared initially. I mean, once again, things have changed. Now you have all kinds of things like material that are shared, reproducible, and so on and so forth. But it's an evolution. It's not something that was present even 10 years ago, 5 years ago. As I mentioned before, replicable doesn't mean valid. We don't have time to go for this, but validity goes beyond replicability. Like in psychometrics, there is a distinction between reliable and valid. Conceptually, it's the same thing. You can have reliable results of an invalid measure. So you can think about replicability. It's better to think about replicability as a continuum. Depends on how many levels of variables you have there. So how many, basically, your differences there are between the originals, because strictly speaking, there is nothing exactly replicable, strictly speaking. For no other reason that participants are changed, time has changed. So if you go for the philosophical, continuous this definition of what is replicable, full replicability, there is no such thing. But there is direct, exact replication because of most relevant things are identical. And then to the other extreme, you go for generalizability, validity, because, for instance, you change the way in which you measure your dependent value, or you change the manipulation. So you have differences, but conceptually, you're trying to look for the same phenomenon. That's very interesting, but it's not replicability. Replicability is more about reproducing things using as similar as possible aspects of the original. The Reproducing project, Brian Nosek has already had a talk. I don't know, of course, he knows, he is the leader on this kind of work and in general the reproducibility. So I'm not gonna go again, repeat the stuff, but just again, the only thing to mention the irony that this has been called reproducibility project, when it should have been called replicability project, to be clear. The bottom line is, you know the way it goes, and basically is that roughly a bit more than 1/3 of the results, depends how you count them, but basically 35–36% of the results replicated, defining replication as a significant result. You have different definitions. One way to define replication is that there was a significant result to the original. There is a significant result. So I replicate the effect. That's not fully convincing. That's not the only way, but still it does capture something relevant. Using this definition is all less than 1/4 of the studies have been replicated, and also other kind of interest. But the most important point, I think, this was a milestone achievement of psychology. Because it's not a problem unique to psychology. I think we have to be very clear on this. The sense of course, yeah, that was the study. When you replicate 40%, it's not just a question of psychology. There are many aspects. I'll just tell you a few things about the recent cancer biology project, in which they tried to reproduce results from clinical trials in cancer biology. Well, the results are pretty much not very comforting in a sense. And once again, you can read them in different ways. These are all kinds of references you can go to if you're interested to deepen up this specific aspect. But the point is that perhaps here is a way to summarize it. First, there is a huge difficulty simply replicating the results. they started with a goal to replicate 193 experiments. They didn't manage to. Spending years of effort in trying to do that, they managed to replicate only 50 for a number of reasons, from lack of details, uncooperative first authors. I mean, basically, if the first author of the original paper, I mean, the main authors of the original paper, helped, the likelihood of doing the study was much larger compared to lack of cooperation. Simply, you didn't know the details, you didn't know essential stuff. So the point is, hey, it's very challenging to replicate, to start with. Second, on those that you manage to replicate, which is a subset. So probably it's an optimistic perspective. Well, I don't want to exaggerate in a pessimistic view, but simply it's one part of the study that you try to replicate. Well, if you look here, again, you can have different definition. The one which is similar to the definition we used before, statistical significant, like in the original study, basically 40% of the results were replicated. We're talking about preclinical cancer biology. I mean, you can think, okay, psychology, who cares? Doesn't really have an impact in your work. Yeah, we are psychologists. Who cares about what you do? Cancer biology. We're talking about something that eventually people can live or die depending on the care that has been established based on the preclinical cancer trial. So, yeah, it's highly consequential. Still, that's the kind of situation. And we have replicability projects going everywhere in the last few years. I mean, the original project by Brian was 2015. Now there have been many more of these. It's just a few examples of the many replicability projects in economics that have been replicated, in experimental philosophy that have been replication, and in social science, and so on and so forth. But also another interesting bit is that if you look around, other sciences are following the example of psychology. So again, that's a very positive development somehow, because other sciences are saying, okay, look, it's not just that psychologists are kind of losers, hopeless guys that they don't find. Well, here is a problem. They are starting to check in their own house what is going wrong. We did not, or we are not yet starting. We should. That's what is coming out. The narrative is going that way in social science, in education science, in chemical research, in environmental modeling, in computer science, in qualitative research, in political science, and macrobiology, neuroscience, and so on and so forth. Biology. So it's many fields are following the example of psychology in terms of checking what is replicable, under what condition things are replicable, and what is replicable. And this is one of the examples of the many replication in psychology. And why I mentioned this, not because I'm particularly interested in the specific text, but because that's, I think, a very important way to understand what is going on when you replicate this text. It's not a black-white picture. It's different degrees of shades. Basically, there are results and effects that are highly replicable. Sometimes with effect size that are even larger than the original effect size. And there are effects that are elusive, difficult to replicate, even by putting many resources. And that's, I think, one of the most important developments that will make this point also later on, that is not simply we have a problem. It's gain in knowledge. People start to get to know more and more what kind of things you can build on, and what kind of things you should be careful. Elusive doesn't mean there is nothing. It means still it's unclear under what conditions something happens. That is difficult to find. It's difficult to build up on that because you don't really know if you're going to get the results under which condition, and so on and so forth. And that's one thing. Another thing is that sometimes you start to discover that most effects get replicated. That's, given the beginning, that's surprising. In the sense of by beginning, I mean the first year of replication, the message out was, it's a mess. We can't trust anything. You can't replicate anything. Exaggeration, over dramatization. But the point is, the negative message was much stronger than the positive message. But then, after years and years, you start to build up, well, hold on, there are effects that are relatively easy to replicate. Just provide a couple of examples because they are giving us some, I think, tips on why possible explanation, of course, it's a speculative hypothesis, possible explanation why they are replicated. In personality, they try to replicate the link between personality traits as predictor of outcomes of a number of outcomes. So, long story short, this is based on many outcomes at the big five kind of standard personality framework, not necessarily the best, but a standard. Bottom line is most effects are replicated, and with effect size that are not too far away from the original. One possible explanation, as somebody who worked in personality can testify firsthand, is when you are working that you don't care really on whether something is significant or not. Everything is significant if you have a large strength. You are focused on the strength and robustness of it. That's your focus. That's your mind where you're focused, not on demonstrating something significant. Most often, you have the opposite problem, to eliminate the fluctuations from the really things that stand out, which means, as a field, the focus is on robust outcomes. So probably that's one reason why it's easier to replicate these results. Cognitive psychology, some classic findings in cognitive psychology, like Simon task, Flacker task, spacing effects, and so on and so forth, have been replicated even under conditions that could have been, kind of challenging. Like, for instance, online instead of in the lab. We know that some kind of cognitive experiments require focus attention, otherwise you don't have the effect. And online is more likely there are disruptions. So you could think, okay, that's a more challenging conditions under which we have it. Still, this is one example. Many effects have been replicated. And guess what? One common thing among these effects, they are all within participant studies. The effect is based on effects within participants. I will elaborate on these 2 points. So, first part, some initial take-home message. Just because something has been shown once, it does not mean it is a consolidated. That's I think the big lesson of the replicability crisis. And there are effects that are robust and relatively easy to replicate, and effects that are difficult. We can call them elusive effects. I hesitate to say no effect, because often it could be also there are conditions in which you have effect, but it's difficult. It's not easy to find. And it seems as if I didn't put too much emphasis in the for question of time, but there are quite a bit also of evidence that the contextual variation, the example before, cognitive experiment online versus lab, it seems to matter less than initially thought, and what matters more is whether you find the effect or not. So contextual variation of this type, like the country or online versus lab, they do not seem to matter as much as whether an effect is robust. You can find it. So, consequences, replicability, as you know, is starting to have a stable place in psychology, not only in psychology. A second important consequence I mentioned also before is that it is a more nuanced understanding of what is going on. There is belief in some effects which is low, but another effect is high because they're replicated. So which means you build confidence in these effects. There are changes in research and publication standards. There is much more awareness of false positive and quasi research practices, more attention to methodological issues, more sophisticated, sensitive approach. There are many positive consequences of the replicability process. Now, after all this, I focus on a few implications of that, I think positive consequences of replicability that has put attention on how to get it right. Because what is the logic? The logic, you need one step to get to right there. The step is, first, as a scientist, most of us, most of the time, we like to get something right. Then, of course, you also like to publish, then also we like also to do anything else. But we like to get something right. There is an epistemic motivation that is really strong in science. It's about knowledge. It's about getting to know stuff. If we get it right, it's going to be replicated or replicated. So the question is, one can rephrase the question, what does it mean to replicate? That goes at the heart of it. Because if you get it right, it's more likely that it's going to be replicated. Again, there is possibility that it's not replicated due to all kinds of things, but it's more likely. And as a kind of generic metaphor of all this generic message, I mean, abstract message, is to increase the signal, decrease the noise. So that's what we should try to do in our studies, in our research. Try to increase the signal, try to decrease the noise. As a consequence, things are going to be more valid, so we get it right. As a consequence, they're going to be more likely. So I'll just do a handful of pointers, a few pointers on errors of statistical input. Start from there, power and precision, and then increase sample size, simple design, and make a quick but clear distinction between parameter estimated statistical input. I'm going to go through the second part through this 5 topics, some in more in depth, some very quick. So let's start with errors of statistical inference. What does it mean, error of statistical inference? Well, it means, in science, you make errors. If you don't want to make errors, change on. In science, you make errors. You have to make errors because it's probabilistic. So the best you can do is to try to understand and to control how many errors you have. And starting from a frequentist approach, one could use a Bayesian approach. And as a matter of fact, there is NHST, null hypothesis significant testing, but there is also now BFST, bias factor significant testing. Same problem, different names, same problem. There is confidence interval, but there is also credible interval. Different names, same problem. There are technical differences. There is a top-down versus bottom-up. Strong philosophical difference. Yet, if you go to the bottom line of it, you make errors in one case, in the other case. And the problem is change name, sometimes put aside, but still the problem is no matter the statistical. I will approach it from a frequentist perspective. From that perspective, you have 3 types of errors. False positive, false negatives, and inaccuracy or imprecision. So the false positive, false negative are linked to the null hypothesis significant test, which is basically what p-value less than 0.05. Wait, what you have been told as a student. That's the kind of null hypothesis significant test. And the imprecision, one, if you're interested in that, which is very interesting, I think I highly recommend this book by Karen Jagman on what they call new statistics, which is not new in the sense, but in some deeper sense it's not new at all. It's refreshing because they focus on uncertainty in the estimate, confidence interval, things like this, which are rather than on whether it's significant or not. So it's a slightly different way of specials. So if you focus on the first one, what is the law? Well, in the frequentist world, you have 2 things. You have what you see and what you don't see. What you see is your sample of data. What you don't see is your population. You are interested in making claims about the population, not your data. You use your data as a way to make claims. And you basically never, almost never, have the luxury to have the population, to analyze the population. You have data. Being a studies, being a survey, whatever it is, you have samples most of the time. Even in the extreme, most of the time you're safe, or not to say all the time, but basically, you have faced with samples, which means you make inferences from the results of the sample, hoping that they're correct also in the population. One way to understand this is significant, not significant. So it's one decision-making system which is attached to basic statistics, as I shall articulate later. There are 2 different things. It's not the same. It's attached to basic statistics and basically allows you to make practical claims like, it is significant. No, it is not significant. I can reject H0. No, I cannot reject it. And by doing that, you can be right or you could be wrong. So you can make 2 types of error. Type 1 and type 2. Type 1 error, you all know this stuff, so I'm repeating it at home, but just to be sure. which are on the same page. Type 1 error is so-called false positive, is when you think something is significant, so you reject null hypothesis. But in fact, the null hypothesis is through the population. You find the significant result is not replicated. That's a false positive. False negative, the opposite problem. The opposite problem in the sense of you don't find something that should have been found because in the population actually is there, but you don't find it under your conditions. That's the false thing. Now, how can you control error? As I mentioned before, you can't avoid error. You can control it. You can accept a certain amount of error from a frequency perspective, which is something like the alpha rate or p-value, which is the how much you accept to be wrong. Suppose you do 100 studies. If you accept a threshold like 0.05, it means that using that threshold to declare significant, 95% of the time you'll be right, 5% of the time you'll be. That's the error. And that's the how to control the type 1 error. Type 1 error is the false positive. 1 tail, 2 tails, you know, whether it's directional or unidirectional, bidirectional. The type 2 error is instead when you don't find something that should have been there. And that's the concept of power. The concept of power, the definition of power is the probability of finding an effect if the effect exists. It's the complement of the error, to be precise. So if error is called beta, power is 1 minus beta. So if you have an error, type 2 error of 0.20, it means you have a power of 0.8, which means, assuming that there is an effect, you have 80% of likelihood to find that effect. So that's basically the kind of logic which is telling you the type 2 error. That's how you control this kind of possibility of error, which brings us to the concept of power. Now, we could have, I'm going to be exceptional, really on the key idea behind this. You can approach it in a different way, more or less formalized. I'd rather go to the key bottom line of it. The definition is this one that we said before. The probability of finding an effect if the effect exists in the population. How much is likely to find it in yourself? That's the definition. Now, how you can estimate power? There are 4 elements in power. The type 1 error or decision criterion, alpha, we mentioned before. How much power you decide, 1 minus beta, we just mentioned. Type 1, type 2 error, right? The sample size and the effect size. The beauty is that fixing one of the elements, you can derive all the other. A simple example here. The beauty is this. The ugly side of power analysis is that there is no such a thing like power. You have all different powers for all different kinds of situations. You have to make calculations depending on the design, level of measurement, the complexity, your hypothesis, how many hypotheses you test, and so on and so on. So it is easy conceptually, quickly becomes difficult, complex in terms of application. But just to focus on the simple example, 2 groups, experimental control. A very simple, clear-cut example. We fix alpha, conventional 0.05. We fix power, conventional 0.80. Now we can have sample size as a function of fxn and everything else. So basically, you can have a curve which is telling us the combination between fx size and sample size. For instance, if we expect an fx size of 0.5, we need a sample size of 125 to add power to have 80% of likelihood to finding that effect, an effect of that effect size, or larger than that, of course, with alpha point equal to 0.05. Now, you see this curve is very straightforward. It means fixing your likelihood, so fixing how much error you want to accept. It means the larger the sample size, the more you are able to detect a small effect, and the other way around. If you have a large effect, even a small sample size can be enough. Metaphorically, imagine you are in a room. If you are searching for a needle, you need to look around for a long time. If you're searching for an elephant, 1 second is enough. You look around, oh, that's the elephant. There you go. If the effect is large, few participants are enough. If the effect is small, you need many participants. Everything else being equal. Okay, and this is the question, this is just for curiosity. When I say they are linked, that's one example of it. So this is alpha, that's beta. This part here represents the effect size expressed in a way that is not that familiar to you, perhaps, but there's nothing else than the effect size. And n is how many participants you need given this. So you input your zetas, your alpha and beta, the zeta correspond to alpha and beta. You say what is your effect size using this metric, and then you derive how many participants you need. That's it. That's how it works. So what affects power? As I mentioned before, metaphorically speaking, if you have an elephant, it's easy. If you have hours to search in a room, it's easier to find something. So either you have a large effect size or you have a large sample size. So the question, when sample size become larger, more participants, like n is increased. But when the effect size is larger, and then we go back to the metaphor of the beginning, signal noise. You have all different ways to express effect size in different methods, depending on condition, but they have this kind of structure is the ratio between the signal and the noise, or the total variation, which includes the noise. So basically, it's how much signal you have given the noise that you have. It's a kind of abstract way of thinking about the effect. And you can have power as a function of effect size, and this is now we are going for 3 different variables, so you can have curves of power. I mean, we are going to pass through that. But simply to say how you can increase power, said before, increase sample size, increase the signal, or decrease the noise. That's it. That's how you can have more power, which means you are more likely to find the effect. You are more likely to find either the needle or the elephant or anything in between. Now, why should you care? If less power would mean only less false negatives, well, unlucky you, it means you find less things. But the point is, power has an indirect effect on what statisticians call crucial error rates, so they have different names, true error rates, meaning that indirectly has an effect also on false, not only on false negatives, directly on false negatives, indirectly on false. And in general, if you have low power in the field, it means that you have a body of complete evidence and you have many false positives. So it's important, as important consequences for power. And everything else being equal, power is affected by the design of your study. So, or as I mentioned, the design, only one point, I want to make only one point more in detail, the other points rapidly. Within subjects are much more powerful than between subjects. I mean, these are a couple of references if you're interested. In general, in power, more specifically, the one by CRISPR for the design. But just to give a sense, very simple situation. Just imagine a very simple design, 2 by 2, mix different types of design. This is what you are focusing on, 5, 5, 5, 6. Imagine you have a situation in which you have a baseline measure, 2 groups, randomly allocated, 5, 5, right? Then we have an intervention. One is a control group, the other is an experimental group. We think that the experimental group is going 1 up, the control group stays at the same 5, 5, 5, 6. It's a very typical situation in which you have a baseline and a measure after the intervention. That's the pattern. Always standard deviation equal to 1, which means like 1D cohorts here, you are basically in this critical condition here. Always we are interested in interaction effect. Always we want to have power of 80, alpha 0.05. To achieve that power in a full between subject, we will need 128 participants. In a within subject, correlation between the variables of 0.50, we will need 10 participants. Dramatic silence, 10 participants. Less than 10 times. It's a huge difference. And remember, I mentioned before the cognitive paradigms, basic cognitive paradigms, they are robust. Well, one of the reasons is that a smaller number of participants can have, given the proper design, can have, if there is a large effect, like a super effect, large effect, you can have a very reliable result. Once you establish, you can trust them. You can find it more likely. So, yeah. So the question becomes how to increase power. I mentioned before, again, now from a practical perspective, increase sample size. That's straightforward and that's always good. If you can, use within subject design, but sometimes you can't. So you have to be very careful because sometimes there are theoretical, conceptual, and practical issues that don't allow you to have within design. Just a very extreme example, if you want to check the efficacy of two different clinical interventions, you can't have the same participant go through both. You have to decide somebody's getting one, someone else is getting another one. You can't have it. The one is going to affect the result, the other comes to the other. That's an example which the within design is not very good. Think about larger effect size, because that's the critical issue again. Think about how can you frame the question such that you look for the elements that are going to be. But of course, you have to be wary of possible reduced ecological value. So you're thinking about it. And then there are a series of other aspects, which bottom line boils down on, one of the bottom lines boils down on reduced denials. Reduce sources of variations in the data, which are not due to the variable of interest. Because once again, as I mentioned before, the general logic behind effect size, you have very different equations, but they have the general logic, signal to noise. Increase the signal, decrease the noise. That's power. But yeah, why should you use power? In fact, it's not the only way to approach stuff. I mentioned before personality, right? I pass most of my life career without having done a single power analysis, because just I couldn't care less if I was significant or not. I would care about, is it sufficiently stable result? Can I trust it? Not what is significant. Okay, obviously significant. But with infinite sample size, everything is significant. Now, in that kind of logic, the question you ask yourself is rather the stability of the precision of the estimate, the accuracy in the estimate. Now, in a precision approach, it's better to use the word precision because accuracy implies that it's valid. Precision doesn't imply that. The point is, in that kind of logic, if you're interested in being precise, sample size is what you need. Because you are not interested in making inferences about that. You're just interested in estimating the value. So if you want to get it right from that perspective, increase sample size. And because precision and power are different aims, the first study and the second study are good from a significance perspective. You know, the brackets means confidence interval. 0 is this line here. It means we can reject 0. Here there is a large confidence interval. Here is a small confidence. Both cases we can reject. Great. But the precision is quite different. So from a significant perspective, 1 and 2 are better than 3 and 4. From a precision perspective, 2 and 4 are better than 1 and 2. From a precision perspective, it's better to be able to be reasonably accurate to say, well, I'm very quite convinced that there is no effect. The effect is equal to 0. So precision is valuable, no matter everything else. And that's why I mentioned this book. There is a minor practical problem is that you need big sample size, no matter your effect. Precision is insensitive to the effect. So we saw before that power changes is a trade-off with effect size. Precision is basically a flat line. Regardless if you have a huge effect size, like, I don't know, a damage in the brain of somebody, if you want to be accurate, you need hundreds or thousands of participants. That's it. The effect size has a minuscule effect. stable estimates. Here, I want to make it very quick. And a stable estimate means basically that the bigger the sample size, the more you can trust on your estimate. I'm going to make just one example of my own data, in which the idea is this, that we calculate the correlation between two every new participant comes in and see what happens over time. Of course, at the end, it is the same as the estimation at the end of the study. So at the end of the study, this was almost 0.05, almost nothing. It's the correlation between two independent personality traits. So I would expect they are not correlated with each other. Makes sense. But we calculated each step. Now, after 40 participants, I can say that this statistically significant, less than 0.001, a positive correlation between the two. If I were to do a sequential study, I would say, stop everything. Huge discovery. We discovered that, unlike what everyone else says, we have a positive correlation between honesty and agreement. The openness, the other one is extroversion. We have a highly positive correlation 0.5, more than 0.5. Big story. Everyone, no, yes, no, new studies and so on. Is sand. The castle will dump on sand and there is water coming. It's evaporating in a second. Again, goes to zero. And we did a study on this. What happens in terms of stability? At the bottom line, if you're interested, you can go there, but the bottom line is that one has to be careful in doing sequential studies for small samples. Very, very careful. Because you can easily end up in a situation which you find which is deceiving. There are proper ways in which you can do sequential studies, but you have to be very careful with the approaches. And there are references if you're interested in developing this. But the basic idea is that estimates stabilize with bigger sample size. Reduce variability, again, increase the signal, decrease the noise. Reduce variability means standard error is the amount of inaccuracy, because confidence interval is roughly twice the standard error. So it's the measure we have in frequentist approach to say the imprecision of the estimate. Now, if you want to be precise, you need to have a smaller standard error. The standard error, as you can see here, depends on n sample size. We already said that, and on s, the variation. Standard deviation of variance. So that's different metrics. The concept is the same. But the point is, which means if there is less variability, if there is reduced variability, you have larger effect size and easier to find the effect and easier to replicate it, which means, unlike what sometimes you have the intuition to go simple, go clean. Because in this way, you reduce sources of variation that are due to other forces, and they are increasing the noise. So try to get the focus on the scene. And one thing which there is no more time to develop here, but it's interesting for those of you who are working more in the, especially for those of you working more in neuroscience and neuropsychology and things, you can, up to a point, trade off trials for participants. So these are a couple of references here, and here there is another one here, but so I'm not going to go into the details, but the bottom line is that depending on the relative variability within participants versus between participants, there are conditions under which you can add, let's say, 200 trials to one participants have reliable, robust results, instead of adding hundreds of participants with few trials each. So basically, the bottom line is that power, robustness, and replicability of the effect depends also, at least in part, under some condition, to have a certain number of trials. So you can trade off trials when having participants is very costly. You could increase the number of trials, whatever kind of paradigm you use, to have more replicable, robust systems. That's one of the tricks of the within subject effects. It's usually based not on a few trials, but a certain a relatively large number of psychophysics, another example. Finally, parameter estimate is different from statistical input. Here, basically, I already said before, but I put it to emphasize it strictly here. What I mean is that, bear in mind, there is a very basic statistical principle. More data are always better than less data. That's it. More data, more sample, more participants, more trials. Because precision matters no matter everything else. That's a basic statistical principle. You have to think, it's better that you think about significant testing, regardless whether you use null hypothesis significant testing or null hypothesis Bayesian testing. Because, so regardless whether you use frequentist approach or Bayesian approach, these are decision-making tools on statistical data. Do not mix them up, the two. These are practical decisions that you make, significant or not significant. More likely one hypothesis than the other one. But the basic statistical principle is there always. This has to do with things like the more data you have, the more your estimate is precise. That's it, it's very simple. And that's, again, precision versus power. So precision is power, no matter everything else. But sometimes you need to make, you have to, it's your duty. So yet, sometimes it's your ethical duty to make allocations of resources such that you don't have huge sample sizes, huge trials. You have to make a trade-off, a compromise. So to wrap it up, design your study with adequate power, because not only you have less negative, false negatives, but also indirectly less false positives. In general, if you have more power, it means you have less overall errors of inputs. If you have the resources, you have the possibility, go for larger set. And or more trials, more items, depending on what you can maximize in your specific community. But more data is always better than less data, because precise estimates are better than imprecise estimates. If you have, I mean, one could make even the philosophical point that asymptotically, it's all about precision, nothing else. Because when you have a precise estimate, obviously, whatever decision you make is correct. The question is, how do you get there? Sometimes you can't get there. You can only have compromises and so on and so forth. So there are a number of reasons why it's important to have decision making, inferential statistics, as it is called. But remember, more precise estimates you have, the less error you make. That's it. That's it. That's it. And if you make less error, you are more likely to get it right. Remember, get it right doesn't mean you are right. As you will painfully discover, sometimes get it right means that you were wrong. Because you discovered that your hypothesis was so cool, so great, but incorrect. And this happens many times. Sorry, it's a very difficult one. Design simple and clean stuff. Get it simple. Reduce unnecessary variability due to many secondary details, and think about the conditions under which the effect size is larger. Increase the signal, reduce the noise. So that's the kind of general message. And remember, you got it. Keep calm, you can do it. You can do it. Thank you.
Okay, so thank you, Professor Perugini. I think we can start with our question and answer session. If anyone has questions, please, let us know in the chat, either by just writing question or writing the question itself so we can read it out. And okay. Christina, is that a raised hand?
Yeah, if no student has questions, I was wondering one aspect. Okay, I understand it is important, of course, the number of trials that you have, but sometimes it depends also on where the noise comes from. I mean, sometimes the noise comes from inter-individual differences. So even if we raise, we have a lot of trials, still it is not enough. I wonder whether you have suggestions for us when it is particularly important to take into consideration the numbers of participants, and not only, yeah, if this general advice is always to be used, or there are specific situations in which it is more important to consider the number of trials or the number of participants. I don't know if you see what I mean.
Yeah, thanks, Christina. It's a very good question. I mean, no, it's not under all conditions. I mean, it's generally speaking, the point is that basically is the assumption is that the more the phenomenon is static across time, the more increasing number of trials is good. Everything else being equal, because basically you're not changing what is going on. Sometimes by repeating trials, you introduce learning, fatigue, lack of motivations, and so you are changing what is going on. And under those conditions, increasing number of trials could even be detrimental rather than incremental. So you really have to be thinking carefully about this. But the reason why I mentioned is because the assumption is that only by increasing number of participants you can have more power, because that's what the standard error is telling you. But sometimes you can do it also by number, by increasing the trials, and also because otherwise it would be almost paradoxical that there are fields like psychophysics, the example, in which they are being constructed on few participants doing thousands of trials, yet some of these effects are very reliable, not all the effects, but some of the effects are very reliable, even though they originated from a few participants doing many, many trials. How's that possible? They were so lucky, they were reliable. No, the question is, in those kind of conditions, probably the noise introduced by the lack of staticity of the phenomenon is lower than the gain introduced by repeating the remeasurement over time. So these are conditions. Of course, ideally, you would like to have many participants and many trials. That's obvious, but the point is that there are trade-offs in which sometimes you could go for more trials rather than more participants. Some other times, it's better to go for more participants and more trials. So depends on a series of aspects, but one has to be, there is no simple unconditional rule. But the point is that both can work. Within, I mean, this is also has been clearer because I think the last 10, 15 years, there's been more emphasis on studies, mixed multi-level studies, in which you can decompose the within participant values from the between participant values. And in many fields, for example, personality, in which the assumption was that, traits are, you are conscientious, and that's it. Then there are differences between people, but basically that's something which doesn't change much. Results have shown that there is quite a bit of variability also within persons, not only among persons, which means you can increase, if you take it from this perspective, you can increase stability of results also by increasing how many times you ask people, and this is one of the reasons there have been big development in that field of the ambulatory assessment. Smartphone many times a day, many questions, because you can achieve this advantage.
I have a slightly more practical maybe question. I'm not sure, but since you mentioned multi-level models, it's quite straightforward to me how to more or less figure out how much power a study can have when the analysis is quite simple, but there's more and more often more complex models or even network analysis. And when I'm faced with trying to figure out how to design one of those or how to figure out how sensitive it is, I don't know what to do anymore. I mean, it becomes very complicated, and I don't know if it's enough to reference something simpler or how to behave.
Yes, this is what I sort of hinted at, that the logic is simple, but then the practicalities are quick to become complicated. Because if the underlying model and the hypothesis is complicated, power, and there are cases in which there is no algorithmic solution to power model. The only thing you can do is simulation. That's it. You can only simulate the models and put what are possible sensible values, guess sensible values, and then, given these guesses, estimate so many participants, trials, whatever is your unit you're interested in, you need to achieve a certain level of power. So, in general, first, there is a development in the last few years, again, of more and more use of simulation as a way to try to solve complex problems.
Okay.
That's great. It introduces another problem that usually in complex models, you need to simulate many parameters. You need to guess many parameters. Some of the parameters are reasonably easy, or at least not that difficult to guess, or at least reasonably difficult to guess, and some are really complicated to guess. You always feel hopeless. How can I know what is this parameter, the value of this parameter? So what starts to be done is something which has pros and cons, but at least when you don't know anything could be useful, is to look for similar studies or even pilot studies for the parameters that you need to fix in the simulation, so that you reduce the complexity and you play only with the key parameters. The second approach, which goes in the exact same logic, is try to formulate the hypothesis, the simplest possible hypothesis among the many that you're interested in, and focus on it.
Okay.
Complex model, but sometimes you can rule that you can reduce a complex model in a simple, more straightforward, specific hypothesis. Focus on that, empower on that, and make sure that you have enough participants who have reasonably stable results, or reasonably. I mean, don't. As I mentioned before, in an ideal world, you wouldn't even. Power is, the kind of. There is a very nice expression by Cohen. Cohen is the one who discovered power. That basically something like, I felt like going to heaven and then to hell and to heaven again. Because the point is that in principle, power is pointless if you have enough resources. Okay. But the problem is that you never have enough resources. So you should care. And that's where the problem comes. The problem comes in this world. In the ideal statistical world, it's all about precision. In this world, you have to make compromises because you don't have the resources to do this. No, you can use them all for once. And then when the concept of power comes from, but the concept of power boils down, if I have to go for something more heuristic rather than the specific calculation, because also you have an additional problem, specific calculation. You have unknown. How do you know what the effect size is going to be? If you would know what the effect size is going to be, you wouldn't do the study. You already know the answer. So if you know the answer, why should you ask the question? You don't need to get this study. So of course, it's always a guess. And as any guess, you can get it wrong. Most of the time, you get it wrong. So all these kind of things. I think it's useful to think in terms of robustness and sensitivity. In terms of, is this design, this experiment, sensitive enough to, am I going for enough time in the room to find the needle? Or I'm trying to find the needle in going or spending 25 seconds in the room. How can I find the needle? So that's sensitivity. It says, do I have enough to find what I'm looking for? Second, robustness. Is it what I'm looking for something that perhaps will come up once, never come up again? It's something robust in the sense of that, do I have, let's say, do I have enough resources to trust my own results? So you can frame, I think the bottom line is, when you publish, you try to publish. And that's how it works. And that should work. But first, you need to convince yourself that what you're trying to publish is good. And then try to convince others, not the other way around. So first, you need to think that what you're doing is correct. Then you can get it right, you can get it. But you have to think that, it's correct. And then you try to, in this logic of trying to get it right, to get it correct, it means also, do you trust your own results? Or do you think this is something that, oh, okay, I got it once, I'm not gonna get it again. Let's publish quickly before somebody discovers.
Okay, thank you. We have one more question again. Francesca is asking if we cannot control the sample size at all, and we need to.
I mean, more reliable measures. That's
okay.
it. Most more reliable measure. One of the ways which you can increase reliability is by increasing trials. If you think about what is reliability, reliability is, internal consistency. Internal consistency is basically Cronbach's alpha, for example, or measure. Cronbach's alpha, the most known internal consistency measure, depends on two things: the average correlation and the number of items. Which means if you have more items, you have more reliable measure. But more items is nothing else than more trials expressed in a different matrix, in a different concept. So I don't see a contradiction between the two that we're saying. To have better measure means to have more reliable measure. To increase the measure means that to have more reliable measure can mean to have more reliable, more trials in the measure, more items. Outside the measure is more trials in the sense of behavioral response. Because the trials if you use the word item, an item could be a behavioral response, or could be an item of. So perhaps item is the kind of more generic term and depends on items. Items could be trials, behavioral trials, some kind of responses of any type of cognitive paradigms, or could be more items in a question to increase the ability of the both work. How much they do work, again, depends on the specific thing you are looking on. That's the question before Christine asked before. It's impossible to make a generalized claim because it depends on the relative ratio between the relevance of the inter-individual difference in respect to the intra-individual difference. So do matter more the differences between people in relation to the difference within people, or the other way around. What is the relation between the two? And in general, for how the question is written for the power analysis, this is a point made by the paper by Rauder, and I don't remember the second author, but it's one of the papers in the slides that I mentioned there. The point is that in general, under all conditions, sample size has a larger effect on power than trials. So increasing participants has a larger effect because it enters in two ways in the equation, the standard equation. But under some conditions, the effect of increasing trials is very large. So you could trade off this. I can think of, for instance, otherwise, people working in field like neuroscience or neuropsychology, you know that you have a kind of fMRI study with hundreds of euros for a single participant. Once you get there, you try to have hundreds of trials to compensate for the cost, rather than having thousands of participants. And of course, now they realize that even in those cases, you need to talk about some kind of effect, you still need hundreds of participants. Otherwise, you have effects that are not going to be there. So they start to realize this. You start to see more and more in that field, like it happened in genetics, that more and more in genetics, they are going on hundreds of thousands of participants. In neuroscience, they are starting to go in hundreds of participants. But before, the magical number was 50. Who knows why? But there was a magical number of 15. You need to have 15 participants for a fMRI study. Until 10 years ago, this was the magical number. Now, 10, 15 years ago, now they start to go for hundreds of participants.
Thank you very much, Professor. Any other questions? Last call? And if not, I think we can close.
It's a very interesting paper. I didn't know.
We have just some reminders before we close then. I think we can share the, sorry, just a second. I think we can share again our closing slides. Okay. So I want to thank once again Professor Perugini for his talk today. That was really, really interesting and useful, I think, for everyone, also on both the more theoretical and more practical implications. And before we close, a few information. First of all, again, an email, everyone who requested a participation certificate will receive an email certifying your attendance to the seminar. It will come within the next 7 days, so please give us some time before inquiring. The emails will be sent in batches, so you may not all get the attendance certificate on the same day. If someone else has received it before you and you haven't received one yet, give it a few days, it should come. And then if you haven't received your certificate within a week, then let us know and we will fix it. Also, the certificate we're sending is an email, again, certifying that you attended. If you need a different kind of document, a PDF or something else, please write us and let us know. Vittorio already sponsored it at the beginning of the talk, but the video of today's seminar will be published on YouTube again in the next few days. If you sign up to ITRN and to the notifications, you will be you receive an email when the video is available. Please feel free to share it, like it, subscribe to the YouTube channel, because again, all of these, the more traffic we have, the more visibility the network gets and the more it grows. And finally, I would like to remind you all of next month's speaker, Felix Schombrand, who will, I think it will be the final seminar of our cycle this year, yes? And it will talk to us about open science, metrics, and career. So once again, thank you, Professor Perugini, and thank you, everyone. We hope to see you all next month.